This is Katie Butts-Dehm, your Bio 110 instructor, and tonight I'm going to talk about the leaf chromatography lab. I already have a video, so I'm not going to do the whole lab, but I do want to go through some areas where people have some issues. So I'm going to share the screen first, and here's some things. So since we're in the spring, in the fall and summer, then I like people to get the leaves from trees, but a lot of people do spinach too because it always works. But you can see the different color pigments of the different trees and, you know, when they're going to turn color. Like they look green from the chlorophyll, but later on they may look orange after so much of the chlorophyll is gone. And then you can see the other colors and pigments that are normally masked from the chlorophyll. So what you're going to do is you're going to get leaves from spinach or 3 vegetables if you want. Maybe beet greens might be nice. Or you're going to get, well, I would do spinach. And actually what I want to look at now instead of this one is this right here. So what you're going to be doing is you're going to be separating the pigments in the leaf. So chlorophyll is your pigment, the main pigment used for photosynthesis, but there's chlorophyll A, there's chlorophyll B, those are green, and then there's pigments that are orange like carotenoids and xanthophylls that are yellow. So you'll look through those. Here's the materials, and I have all the materials out, but again, I'm not going to go through these. The one thing that's not on here that I should have had is it might be good to have a cutting board and a knife. So you're going to collect your leaves and let's see, I'm going to go over here. And what I have, oh, I have to unblur my background. I just got spinach and I got the least expensive spinach. And I would use it, you know, cut it all, and then I would use 3 of the amounts. And I have some bowls here. And I've got larger bowls, okay? And the larger bowls, I'm going to put some hot water. And I actually have hot water in the next room. That way you can keep the water heated. The smaller bowls, what I would do, since whatever I use, I would use the same amount. And like I use 3.6 grams of leaves in each container. I would take and chop as much spinach as you can fit or whatever you're using and put the same amount into each of the 3 bowls. Then you're going to pour the 70% isopropyl alcohol, just enough to cover the leaves. And I used 20 milliliters of alcohol. And then you're going to put warm water into your glass or metal bowls. Now, the thing about this is, and actually I should have put cling wrap, I should have had that. But what you're going to do is you're going to take the small bowls with the spinach, chopped up spinach, and the alcohol, and then you're going to put them in the large bowl of water. And make sure that your water's not going to go above the small bowl and into it before you start putting spinach and alcohol. What you might want to do is just take an empty bowl and have this maybe with water and then fill it and then take the bowl out and maybe mark it. And then you know how much is in all 3 bowls. Or cover your spinach and your saran wrap, spinach and alcohol with saran wrap, put it in here and pour just enough to be really careful, like off the side of water, to make sure it doesn't spill in or go over, and then put the same amount of water in the other bowls. Now, the trick is you're going to have the bowl of isopropyl alcohol sitting there and being warm until a lot of the chlorophyll leaches out of it. So you're going to have to rewarm the water several times. And you don't want to put the isopropyl alcohol and the spinach in the microwave. You don't want to heat that up. So you can either take the bowl out with the spinach, the small bowl, and reheat the water in the microwave, or you can just dump the cold water out in the sink and pour warmer water back in and then put the spinach back in there, because you don't want to be heating alcohol up on the microwave or in the stove. You're only heating the water. So you're going to do this until the alcohol is going to turn pretty green, okay? And you'll notice it's going to turn green and then it's going to stop getting greener. It's not going to matter if you leave this in like, oh, I left it in an extra 10 minutes. I don't have the time written there. It may depend on the type of leaves you're using. Spinach usually goes pretty quick, but so there you go. Now you're going to wear gloves, but it's to protect the chromatography paper. So don't handle the chromatography paper without gloves. And let's see, I'll switch this here. So the chromatography paper is white paper that's in here. And I changed from using, like I used to just, I don't know, put the pH paper and chromatography paper different, but people kept accidentally using the chromatography paper for pH paper. So it's very clearly labeled as chromatography paper. And you're going to take the alcohol. Let's see. Actually, before you do that, and mostly I'm going to go through the data now. I just want to get my gloves on so I don't get my finger greases all over the paper. But, and make sure your area is clean. I would normally put it on a paper towel or paper plate or plate, but you won't see it. Everything I have here is white, and so is the chromatography paper. So I did clean my area. And it's just regular paper here. And if you, well, regular chromatography paper. Honestly, with this, it actually will work on a coffee filter. So if you're doing this with kids, you can take white coffee paper, you can cut strips. You can also get chromatography paper off Amazon. I have found that sometimes that using 70% isopropyl, then it actually works maybe even better sometimes on the coffee filter. So if you want to have an extra cup, you can try it with a coffee filter or one of your 3 tries. So we're doing like 3 times, whether you use 1 plant or 3 plants, you're making 3 chromatography papers. So make sure everyone does 3 chromatography papers. Don't just go, well, I'm doing spinach, I'm just doing 1. Okay.
Mm-hmm.
So what you are going to do is, I have so many things out on the counter now, but so you're going to have the spinach pigment with the alcohol. You're going to fill it up to about toward the bottom, okay, on each one. And you're going to want the chromatography paper suspended to where it's going to dip in that bottom. And I don't know how well you can see this, but what you'll do is take it and tape it on a pencil. Now you can use any type of pencil, pen, anything you want to use, scissors to hang it over here, you can do that. But the pencil works better if it has sides so it doesn't just keep rolling. Sometimes I haven't had one that has sides. And let's see. We're going to want it to be about like there. So you don't want it to touch the bottom, but you do want it to, don't want it to touch the bottom, but you do want to dip into your liquid. So that actually, I should have had a bigger piece of tape there. Oh, that's going to be maybe good. Here, let's go a little bit lower. Okay, and it's just not straight. So once you get your first one down, you have an idea about the other one. And you may have a different cup too that might work a little better. But that is a little bit, this will probably work. Oh, I know what happened this time. I was wondering why I need so much tape. It's because these are deeper cups. Sometimes I have shorter cups. So it's not reaching the bottom. Well, that's hitting the side. You actually don't want it to be hitting the side either, but it's almost touching the bottom. Okay, so see there, now it looks good. Can you see that, student?
Yes, I can. That looks
Okay.
good.
Yeah, not
Thank
touching
you.
the bottom. You're welcome. And then you make sure it's not curled, too much. And I'm wearing gloves, so I can do this. Try not to touch your gloves over everything else either, but there you go. So sometimes if you make it too long, you can just roll it along the tape. And again, you don't want it to be touching the side of the cup. I almost feel like, no, that one looks perfect. No. So now it looks perfect. See, it's not touching the sides, it's touching the bottom. I almost wonder about, instead of pouring it into the cup, maybe getting everything set up and using a dropper. And then you're gonna label
That
your,
sounds like a good idea.
yeah, I think that's gonna work better. So you're gonna do this with three papers, and you have an extra paper too. And then you're gonna let it run. Now, what I've found, and this is, let's see, when you do this, usually in the lab, then what you'll do is you'll have this really toxic solvent with ether and stuff like that, and you're in a hood and stuff like that. Well, we're using 70% isopropyl alcohol. You're not going to want to sit and inhale the fumes over it or anything like that, but it's rubbing alcohol. They sell 90%, 91% also, but don't sit over it breathing the fumes. But the thing about it is you're going to have the pigments going to run up the paper. Okay. And I don't have a specific time because it's it depends, it depends on the humidity of the room, the solubility of the pigment, different things like that. So here it is right here, tree one, and see the pencil over here. And there's the chromatography paper. It's taped on the pencil, the bottom's suspended here. And when it sits in this alcohol in the pigment, then you're going to have the pigments are going to be dissolved in the alcohol, and they're going to move up the chromatography paper. And the pigments that are more soluble are going to move faster, so they're going to go further. And also, if they are smaller, a lot of times they'll move further. But you don't want to have your pigment run off the paper. Um, I let my chromatography paper here sit once for hours just to see how long it would take, and it never ran off it. And I think it might just be because it's 91% alcohol and not something, but just keep an
Oh,
eye on it. It may be done in half hour.
okay.
Yeah, so parts of it, here's the leaves when I did trees. They're cut, and it said to tear them, but you can tear them, you can cut them. Here's my small bowls with the water, and not water, sorry. Here's my small bowls with a leaf, and here they are with rubbing alcohol, the same amount in each one, depends on your bowl size. And then don't heat up the alcohol, but you put the small bowl into larger bowls of warm water. And here they are, the small bowls into larger bowls of warm water. And see, there's some saran wrap or cling wrap on there. Oh, and there's my old
Yes.
trivet. My trivet. I just gave that to someone last weekend. That trivet for like over 30 years. Anyway,
Oh,
yeah. So then when it cools, just take the leaves out and add more warm water, dump out the cold, add warm or heat up and then here you have it. The I don't know. But here's your cups. They're labeled. And I'm doing spinach 1, spinach 2, and spinach 3. But if you have a lot of greens that you're going to make out of salad, again, don't use them for the lab and put them back. But you can take the amount that you might use and put that amount into your lab space. And then you might want to do 3 different vegetables because you don't want to have to throw away some vegetables. But once it goes into the lab space, don't put it back with food. Here it
yes.
is. And look, not hitting the bottom, not hitting the sides. And again, I thought getting one of your droppers maybe and pouring it on the side might be better because it's hard to not hit that on the side. And then here it's going to start running up the running
Oh,
up.
that's cool.
Yeah, and see, I had pens before. I'm so glad I had pencils with edges because the pens roll around a lot. And this is just from leaves and isopropyl alcohol. And so you're going to run and you're going to have these parts, and people always have, not always, and some people don't, but there's always people that have issues with knowing what is what. So right here, see where the alcohol ends? That is your solvent front. And mark the
Okay.
solvent front with a pencil, because once the alcohol dries up, it's not going to show. So I'll tell you
Yeah.
again, mark the alcohol front with a pencil while it's still wet. Mark the alcohol front with a pencil while it's still wet. So I just said that 3 times to make sure everyone who watches this
Yes.
video, hopefully everyone do that, because you're going to want to take a picture of this, and I need to see the solvent front, and you can't do the calculations without the solvent front.
Okay.
The other thing, see this, I only have 2 bands here. So from those, sometimes you'll have a band up here. Sometimes your chlorophyll will split. So all I've got is chlorophyll and xanthophyll. So it depends. And if you bump the table that your stuff is on while it's running, then you'll get like a big mess sort of. And it maybe will go up in the middle or something like that. So try to not bump it or not touch it. Just leave, and then every now and then look at it.
Mm-hmm.
So you're going to mark also with a pencil the middle of your bands. And then you want to calculate the RF factor of your pigments. And this is how to do the RF factor. It's RF is the distance traveled by the substance divided by the distance traveled by the solvent front. That's why you need to mark the solvent front. And
Mm-hmm.
it's always going to be the bigger number on the bottom, except for, oh, I'll have to pull out the answer sheet for the table. There's one, you'll get the RF value of the solvent front, and that's going to be 1 because it's the same number over the same number. But
Yes.
so like this, it's not showing everything at once, so I have to make this little. And there it is. Okay. So can you still see this okay?
Yes. Yeah,
Great.
that's great.
Great. So right here, the RF value of my xanthophyll, and here's the ruler. So you just measure from the very bottom of your chromatograph up to midway through the xanthophyll band. And I've got 2.1 centimeters. And make sure that you check, if you're using millimeters, that's okay, but you have to use millimeters top and bottom because the units will cancel out. So you should get the same RF value, whether you do millimeters, inches, centimeters, your units cancel out. But so the xanthophyll, 2.1 centimeters over, here's our solvent front, 5.8, sorry, not millimeters, centimeters. I don't remember what I said, but 5.8, 2.1 over 5.8 centimeters. Your units cancel out because they're the same thing over the same thing as 1. And this is what you get, 0.36. Now the chlorophyll, it's 1.6 centimeters because I measured that from 0 to 1.6 right in the middle of the green band. And then the distance
Yes.
traveled by solution from here to the end of the solvent front, which is why you need to mark it before it dries. And
Yes.
even if you forgot to mark it, take a picture of it, and you can figure this out from a picture, just like I'm showing you. But notice the chlorophyll band, the RF value is smaller than the RF value of the xanthophyll because
Yes.
it didn't travel as far. So smaller number over a larger number. So the closer it is to the bottom, the smaller the RF value, and the more it is to the top, then the bigger the RF value. So here's some more. Look, sometimes it looks like they bumped it, and it did this right here because otherwise it would be flat. And I wonder why the carotene is flat. So look, they have chlorophyll A, B, and then carotene, and where's the xanthophyll? Xanthophyll's right below the carotene, but here it's different. Look, here's the RF values, and look at the carotene. So they're it's right up close to the end of the solvent front, so it's nearly 1.
Wow.
These ones, some of them, sometimes they just make an arc. Oh, because it started with a circle. So you can start with a little bitty glass straw, like a hematocrit tube, just make sure there's no heparin in it. And you can like put a dot and let it dry. And you do that like 20 times for like an hour. and then you run it. And then sometimes you'll get it more with dots, but still you go to the middle of it and you can tell there's two different colors of green. Actually, I see it right here. But and then here's something just showing the solvent front was 16.5. So if you were to figure things out, chlorophyll B, chlorophyll A, xanthophyll, and carotene. Usually xanthophyll's more yellow and carotene's more orange. That looks kind of peachy.
Yes.
I don't know what else I need to show you. This is just more examples. But this one is not figured out. It's just showing you the measurement. And chlorophyll A is always more of a blue-green and chlorophyll B is more of a yellow-green. Honestly, I don't know that you'll get that separation of those because we're using isopropyl alcohol.
Oh.
Yeah.
What would you have to use to get that separation?
I don't remember exactly. I'm thinking something like ether and like a chloroform. I don't, but it's like a much stronger solvent. You do it in the lab, you use a hood, it can pull it right
Oh,
up, and then a longer sheet of paper. It's nothing that you wouldn't do it at home to get such a straight line. And I'll tell you, chromatography is so neat because you can not only separate these pigments, but you can actually, I knew this chemistry teacher who would get this big column of sand, and then he would mix together like different food colorings and then with water and run it through the top, and they'd come out at different levels based on how big the molecule was to make the food coloring.
wow.
Yeah, or you can do gel electrophoresis where you're running little fragments of DNA through a gel and use an electrical current, and they separate um, based on size and solubility as well. But it's through a gel and using a current. But still, you can tell, you know, different type of organisms have a different pattern of DNA fragments. So, but it's a similar, principle that you're using to separate the DNA fragments based on size.
That's so
So
cool.
it is really neat. Um, anyway, so one thing that I do want to show you is, and before I move on to the answer sheet, do you have any questions about all this? Do you understand it pretty well?
I do understand it. Thank you, because you
Oh,
taught it well. I understand.
oh, you're welcome. Thank you.
You're
So
welcome.
I want to look at the data or on the answer sheet, and I wonder if I have any data already, lab supply list, answer sheet. You're going to have this table, okay?
Yes.
And this is where people get, I guess, mixed up. So
Mhm.
if you have, again, rate of flow is RF. I think I forgot to mention that, rate of flow. But so your solvent, the distance moved, and your rate of flow for the solvent's always going to be 1 because the distance moved by the solvent divided by the distance moved by the solvent, right? That's always going to be
Yes.
1. And I have
Yes.
millimeters here and I had centimeters in the other one. It doesn't really matter just if it's like 1.2 centimeters, that would be like what, 12 millimeters? And then divided by maybe 5 centimeters, it'd be like 50 millimeters. Just use the same units on the top as you do on the bottom, and that they'll cancel
Okay.
out. Okay, so the solvent's always 1. Some people are going to have carotene and some people aren't. If you don't have it, put an N/A for not applies or put a dash through it or something if you're writing on it with your hand. But
Okay.
most people will probably have 2 bands. Depending on what you use, some people might have more, but most people will have 2 bands. And so NA's on everything else. And I'll be able to tell pretty easy that it should be less than 1. And then the one that goes the furthest should be larger. The RF value of that should be larger than the one that goes smaller. So if it runs
Yes.
about half the distance, it should be, 0.5 because it would have ran about 50%. So keep that in
Mhm.
mind. And you have 3
Mhm.
chromatograms for tables from your
Mhm.
chromatographs, but you have 3 of them. You need to make sure that you have 3 tables filled out and pictures of 3 different chromatograms, not just like one. Even if you do the spinach, you're doing it 3 times. You can do all 3 at once, so it's not going to take you really much longer, but you need to have
Yes.
3 different ones. And your rate of flow should always be 1. Does that make sense?
Yes, it does.
Are there any questions before we end this?
No.
I'm looking at this. The stationary phase is the phase that doesn't move, the part that doesn't move. The mobile phase is the part that does move. And we're talking about the solvent and the paper. Okay. So the solvent is the isopropyl alcohol, and the paper is the chromatography paper. So it's like which one moves and which one stays still. A lot of people have a problem with that. And before you look at the spinach, when I'm asking, is there evidence that there's more than one pigment before you run that chromatogram? I mean, look, it looks just like green spinach. All you see is green, right?
Yes.
Yeah, so really there's not. After you do the chromatography, you will see anywhere between, 2 and 4 bands, I would think. You could see chlorophyll A, B, xanthophyll, and carotene, but if you see 2 bands, consider that good,
Right.
Okay. So I think but it's kind of neat if you have a tree and you know that it turns colors in the fall, you might want to do this on the tree leaves oh, wait, it's spring, sorry. Just ignore
Would
that.
one that's on the ground work?
If it's a green leaf that's fresh, it probably would, but honestly, right now from where we're at, I forgot that, we're spring, and I used
Yes.
to have this one pull plants all the way to the end in the spring, and then people would have to wait to do all this, but it really doesn't make sense. And then every semester, everything gets moved around. So, and then, most people do the spinach anyway. Because it usually turns out better and you can always get spinach. But if you're sitting around in the summer, you're still going to have everything, and you can go out
Yes.
and get some leaves that are fresh from your tree, pick them right off the tree. And if you're like, oh, I know this tree turns yellow, or this tree is going to turn red in the fall, then you should be able to do chromatography on that and see, and be able to see the different pigments that normally are masked by the chlorophyll. So,
Yeah.
and
Like I can't remember what the red, it begins with an A, but that red pigment.
you have anthocyanins.
Yes.
Um,
Yeah.
yeah, but yeah, you should be able to see all of the pigments. You have carotenoids, you have anthocyanins, you have the xanthophylls, chlorophylls. There's probably even some more depending on it. I've always thought about, well, why don't I do some beet greens and see, because you don't want to just do the beets and see only red, but it might be neat to mix together some things that maybe even some food coloring and see what it
Would
does.
be very interesting because I grow beets and grow radishes and stuff and see, well, is there a lot of pigment in the leaves themselves? You
Right.
know?
So in the summer, then when you have the leaves for those, or whenever you're harvesting them, then you may want to go ahead and use them, and you can just take some white filter, some coffee filter. You don't even have to use chromatography paper, but
Yeah.
just try not to handle it because it gets fingerprints on it, and then it like interferes with the banding and stuff. It's not going to hurt your hands or anything. Also, there's safety glasses, so you don't want to splash isopropyl alcohol in your eyes. And what some people do, if they wear glasses, they'll either put them over them, or some people even have little side shields that you can get and put them on the side of your glasses. So
That's good.
yeah, I actually had, and I got them off Amazon or somewhere, some like readers that were readers but safety glasses, and they were not very expensive at all. I don't know where they are now, but I used to use those quite a bit. But
Yeah.
so I'm going to stop recording soon. One other message, though, I want to mention real quick is midterms are coming up. So, your next exam is going to be on a Friday instead of a Sunday. And I don't like to do this because I know that you have the weekend going on and a lot of people work. So I try not to do that, and I'm looking, but midterms are due. I don't have my screen shared anymore, so I pulled up my computer calendar off the side, but the 8th of March, that is when midterm grades are due. So you want 2 exams on that. So make sure, and also we have labs due like on Sunday and Monday. And there's going to be 2 labs due, but there's been several people to turn in most of those anyway. One of those is the leaf chromatography, and another was a seed germination, which people have turned in that already too, because you just have to wait for the seeds to grow. It's only the lab part, putting it together, only takes about 15-20 minutes. It's waiting and looking at them each day. But so that's why I have those 2 are due near each other. The leaf chromatography, it's not, I don't think it's that complicated. It doesn't take more than a regular lab, and you don't have to do really any prep work other than getting the supplies together. But it does take time to let the chromatography paper run. But please keep that in mind as you're studying. Don't wait till the night before to study for your next exam. You should be studying every day or every other day. Remember to focus on the review sheets, that material. Make sure you understand the notes and you've seen the lecture and everything, but focus on the review sheets and the quizzes, because I take questions directly from the quizzes, and any of the material on my exams can be found on the review sheets and the quizzes. And then I would take each quiz 3 times if you remember, because, or if you have time, because again, some of my questions come directly from the quizzes, and it's just a good way to practice and make sure that you're good with the material, kind of gauge where you're at. So any more questions before we end this?
No, thank you.
Oh,
Thank you
you're
for
welcome.
time and help.
You're welcome. Thank you for coming. And I'm going to stop the recording, and thank you all. Hope this helps you. Have a great day.